This is a production of Cornell University.
Started then. So I'm going to be talking to you all today about a project I'm working on trying to understand how proteins adapt to high temperatures. And we know that temperature is important and it affects both macro phenotypes like plant yield and molecular characteristics of a cell. So for example, we know that biochemistry speeds up at high temperatures, cell membranes become more fluid, and protein structure and composition adjust as well because proteins need to maintain more hydrogen bonds to maintain their structure. And this matters in the context of plants and plant breeding because the world is starting to warm up, and as different areas of the world increase in temperature, we're likely to see a reduction in crop performance. And ideally, what we could do is build genomic selection models to help breeders be selecting for crops that are still efficient at these elevated temperatures. But from a protein perspective, this is fairly difficult. There's a number of roadblocks preventing us from doing this right now. And probably the most significant roadblock is just that we don't actually understand right now which residues matter within a protein. And because in a plant there could be thousands, if not millions of sites that actually matter for protein stability and thermal adaptation, this becomes a fairly daunting question to look at. And that leads to my research goals, which are actually to use a different system, in my case, prokaryotes, to be trying to identify temperature-sensitive sites within the genome and really be understanding the biochemistry behind these sites to see what changes are making proteins thermotolerant, and can we rank or prioritize these temperature-sensitive sites and ultimately decide what do we want to incorporate in these future GS models. Now, I'm doing this in prokaryotes because they make a really good system for studying molecular adaptation. There's a number of reasons for that. You can see 6 of them on the screen here, and I just want to highlight 2 of them. The first is this extreme temperature box. So we know that prokaryotes can exist at 0 degrees Celsius and live and grow at every temperature through and even above 100 degrees Celsius. So there's quite a number of temperatures that they're able to adapt to. And they're also useful because they tend to have large effective population sizes and fairly rapid generation time. And that's important because it means that selection can act fairly efficiently on every individual protein to adjust and optimize its function at that prokaryote optimal temperature. Now, the other thing I need to be able to look at protein adaptation in prokaryotes is an understanding of how prokaryotes are growing and what their optimal growth temperatures are. So a lot of you probably know Emre Çimen. He is a former postdoc in the Buckler lab, and he and I built a model that uses tRNA sequence from prokaryote genomes and uses that to predict optimal growth temperature, or OGT. And you can see from this plot here on the left that this works in both archaea and bacteria and is fairly accurate. And what's really important about this model is that it gives us the ability to predict optimal growth temperature for any new prokaryote species we have, and really all we need is a genome to do that. The other thing that's really nice about this model is that these predictions are independent of protein composition and other protein features, so I can actually use these optimal growth temperatures in my questions looking at protein adaptation. So we're currently in the process of writing up this paper, but for now, I'm using this model to predict optimal temperatures across almost 5,000 different prokaryote species. And so that brings me to my current question about protein adaptation, and I have 3 main hypotheses that I'm focused on right now. The first is fairly straightforward. It's just that protein composition is affected by temperature. And I'm actually not going to get into this in a lot of detail. This is fairly well documented already, and we know that there are some amino acids that tend to shift and become more prevalent at high temperatures. So I've checked this in the data set that I'm working on, and what I see is fairly consistent with the literature. We tend to see more glutamic acid and valine and isoleucine and less glycine. So that lets me move on to my second hypothesis, which is that thermal adaptation is highly polygenic. So this should seem fairly intuitive to you. I've already talked about some of the ways that temperature affects cell function. And so if temperature is affecting every protein, we would expect to see changes in just about every protein across the genome. And so to try and get an idea of which proteins are important or which residues within those proteins seem to be adapting to temperature, I've developed a GWAS model to associate specific protein domain residues with optimal growth temperature of a prokaryote. And the way I do this is sort of like this. I take a set of species proteomes from ensemble bacteria, and that includes both bacteria and archaea species. And I identify protein domains using the PFAM protein database and the hidden Markov models in that database. So for example, if I take this blue proteome, I can find 3 instances of protein domain 1 and 2 instances of protein domain 2. Then I do a multiple sequence alignment for all of those domain observations, and I numerically recode that protein domain just so that it's easier to work with a linear regression. And then last, I just associate those residues with optimal temperature. Now, when I do this, I get a Manhattan plot. So on the left here, you see an example Manhattan plot from one protein domain. The x-axis shows positions in the protein domain alignment. The y-axis is our significance. And that red dotted line is the Bonferroni significance threshold. And so what might pop out to you here is that there are a number of sites that show up as significant in this example. And what's kind of fun about this is we can actually look at the biochemistry and understand what's going on at specific sites. So for example, at this blue site, we see that we can look at amino acid hydrophilicity. And while it's not a perfect correlation, in general, we see that species with higher optimal temperatures tend to have amino acids at this site with low hydrophilicity, whereas species with moderate optimal temperatures tend to have amino acids with high hydrophilicity. And this makes a lot of sense and matches our expectation because we already know that proteins tend to be less hydrophilic at high temperatures. Now, when I do this across all of the protein domains I'm looking at, I see that there are a lot of domains with a lot of significant sites. Essentially, most of the domains I've looked at have enrichments for temperature associations, about 90% of the archaea domains and about 80% of the bacteria. And on average, about a quarter of each of these protein domains seems to be significantly associated with temperature. So that seems to confirm my second hypothesis. Essentially, thermal adaptation does seem to be highly polygenic, and on average, about a quarter of the domain of every protein domain seems to be associated with temperature. And so that brings me to my third question or hypothesis, which is that genomes have a similar evolutionary response to temperature. And what I mean by this is that there are likely to be some key sites that transfer across phylogenetic groups. So I think that there will be some sites that show up as important in bacteria that also then end up being important in archaea. And these sites are particularly interesting because they're already conserved across phylogenetic groups, and that makes them good candidates for us when we're thinking about trying to extend this model beyond prokaryotes and to eukaryotes. And so that's what I'm working on right now, is really trying to identify these sites that are significant across phylogenetic groups. And unfortunately, it gets quite complicated very quickly. There are a lot of sites that show up as significant. And unfortunately, a lot of the sites showing up as significant in archaea are also significant in bacteria. So I've just got an example of 2 protein domains here. We have domain 4 and 118. And what I'm showing you is a blue circle that shows you how many sites are significant in bacteria, and a red circle that shows you how many are significant in archaea. Now, when we overlap these, we get that pinkish purple color. And you can see that focusing only on the sites significant in both of these phylogenetic groups does reduce our search space, but it still leaves us with a lot of sites to be looking at. And so we need better ways to prioritize these sites. And unfortunately, the rank correlation between these overlapping sites is fairly poor. And so we're not able to just prioritize these sites based on their level of significance. So this third hypothesis is very much still in progress. It's true that many sites appear significant across phylogenetic groups, but how we're going to prioritize these is still fairly unclear. And that brings me back essentially to the beginning. So I have my research goal is to be identifying these temperature sensitive protein residues. And I've told you a little bit about how I've started to do that. I'm continuing now to try and reduce this search space. and predict which residues are likely to matter. And I really want to be using the underlying biochemistry to do this. And ultimately, as I've said, I'd like to be building models that can transfer across phylogenetic groups, first across bacteria and archaea, and then ultimately to eukaryotes, and hopefully to plant proteins, so we can understand how plant proteins adapt to temperature as well. But that obviously is not done yet and will be hopefully the subject of a future talk. But I'm about out of time, so I will wrap up quickly, say thank you to everybody in the Buckler Lab for past and ongoing conversations on this project, and I'm happy to take questions or comments if you guys have them.
Well, thank you, Sarah. While people are maybe taking some time to write up some questions in the chat, I'm going to post the evaluation links here.
Okay, and I see that John Luke has asked, in GWAS you have to control for structure. How do you do that in your adaptation? It's a really good question. There's a couple ways that I've tried to control for structure. So one of the ways is to build a kinship matrix based on the Kmer distance matrix from these species genomes. And another way that I've tried to control for structure is using PCs on the domains themselves to try and account for some of the sequence similarity within those domains. Do either of those work well or change the outcome? So using the distance matrix based on Kmer frequency doesn't really seem to change the outcome much. Using PCs does seem to change it if I use information about the sequence directly. Does that answer your question? I'm not sure which Nicholas that is. Rebecca asked, do I have plans to test key loci experimentally? I do not. Any collaborators in line for that? There are some people that work on directed evolution in Chlamydomonas, which would be kind of cool, but I haven't tried to set anything up or really thought about what that would look like. John Luke, I think that temperature adaptation is highly polygenic, to answer your question, but I do still want to reduce the number of loci that I'm looking at, because it would be nice to prioritize the sites in such a way that we can extend this more easily to eukaryotes, if that makes sense. I think that there's going to be a lot of sites changing in bacteria and archaea because of phylogeny or because for other reasons that aren't necessarily going to be interesting in eukaryotes. And so reducing the search space, or at least trying to understand some of the biochemistry underlying the sites that show up as significant, I think will be what's really important to extending the model. John Luke asked, how many loci do I ideally want to end up with? That's a good question, and I don't know. Right now I'm focusing on transferability between bacteria and archaea, and I haven't thought very much about sort of a target number of loci.
Okay, if there are no further questions, thank you very much, Sarah.
Thank you.
Here are
This has been a production of Cornell University, on the web at cornell.edu.